Welcome to our new tutorial. Today we would like to go into further detail about the model geometry and vegetation in ENVI-met. Vegetation is able to mitigate the effects of the urban heat island by increasing the latent heat flux through evapotranspiration, resulting in lower air temperatures. Additionally, plants cast shade on buildings and other artificial surfaces that otherwise would heat up and store more energy. To determine these beneficial effects in urban areas and to evaluate the simultaneous feedback effects of the microclimate onto vegetation, for example during heat waves, ENVI-met is able to simulate the complex microclimate requirements and interactions of plants within their environment. In ENVI-met, plants are objects and consist, like all the other elements, out of grids. ENVI-met allows us to estimate the environmental stress of plants, to simulate the plant's behavior on its surroundings, and to assess its health in an urban microclimate. There are two types of vegetation: plants that only feature a simple vertical structure, which means they do not have overhanging canopies, like grass, corn, or hedges, are modeled as so-called simple plants. The leaf area density, or LAD, of simple plants is represented by 10 layers in the atmosphere, while the roots consist of 10 layers of root area density, or RAD, in the soil model. Plants with a more complex canopy, like trees, are represented by clusters of cells that have an LAD, providing a three-dimensional shape of the plant. Because these plants are carrying their own three-dimensional geometry, they are called 3D plants and are managed by a special editor program called Albero. To examine the processes of trees as a whole, the calculated effects at the leaf scale need to be integrated to the plant level. The total sum of the tree canopy's transpiration is calculated out of the leaf area density of every grid, the local transpiration flux at the leaf scale, and the density of the air in the three-dimensional space of the tree crown. In a case where tree crowns overlap with crowns of other trees, which would result in multiple occupancy of the same grid cells, the LAD cells with the higher LAD take the grid cell, while still belonging to the corresponding tree. The losing LAD grid cells are subsequently removed from the other tree. Besides the leaf model, there is also a three-dimensional root model that integrates all processes of the plant, making a 3D plant a complete organism. In ENVI-met, the amount of water uptake through the roots of a plant is determined by the transpiration of the plant. The transpired water, regulated by Jacob's AGS stomata model, is extracted from the soil and occupied by the roots of the plant. The vertical distribution of the roots is determined by a one-dimensional profile of root area density reaching from the surface to the lowest part of the root. To enable vertical distribution patterns, the root profile is subdivided into 10 equidistant layers. The horizontal extension is defined by a radius around the tree's definition point, which is normally equal to the location of the stem. Out of the root's geometry, the actual amount of root surface area in each layer is calculated by the root area density of the layer and its horizontal extent. The root's radius can be adjusted individually for each of the root's 10 vertical layers. Since absolute values of the root area density are almost impossible to acquire, the root area density of a layer represents nothing but a weighting factor to calculate the share of root surface area for each vertical layer. The extraction of water from the soil model due to transpiration in the tree's leaves depends on the share of root surface area, weighted with the water availability in the soil. Thank you for watching. There are more videos online to learn about ENVI-met.